My name is David. I'm a PhD student from SITS Lab, and my background is probably a bit different than yours because I studied biology initially, and then I went to industry and worked for a couple of years as a wet lab protein engineer. And then I did a second master's degree in data science before I came back to ETH to join SITS Lab as a PhD. And I'm mainly working on machine learning for drug discovery and generative AI for drug discovery as well. And I will talk today about some applications of AI in chemistry and biology. This is a super dynamic field. It has experienced a couple of breakthroughs in the recent years. And I will just try to give you an overview over the current state of the field, what is happening, what are the models that are most frequently used. But of course, this will be by no means an exhaustive overview over the field because it will be focused on structural biology, protein folding, and drug discovery, also drug design. But of course, that leaves out many applications. And also, if I show you an example for a model, this might not be necessarily because it's the best model there is, but maybe because it's the most instructive one, or maybe the first one that existed of its kind. So in these cases, I've usually added these beyond today's scope slides that are just for those who are interested, pointing to other models that are maybe newer, maybe more sophisticated, more successful. Yes. So this is the outline of today. At the beginning, I will just give you a short introduction to proteins and small molecules that we are all on the same page. And then I will transition to the first kind of machine learning model, those that are based on sequence representations of proteins and small molecules. Then I would also like to speak about structure-based models that use 3D structures. But before I do that, I will introduce structure prediction models such as AlphaFold 2 and ESM. And then probably next week, I will mention or talk a bit about some challenges of training machine learning models on biological data, and then start the whole chapter about generative models for de novo design of proteins and small molecules. If you have any questions, generally just raise your hand or write into the chat. That's no problem. So in the first section of the talk, I will just give this short introduction to proteins and small molecules because I think many of you don't have a biology background at all. And we need these basics for all the models that come later. So small molecules are organic compounds that have usually less than 100 atoms. So they are quite small, and because of this, they can diffuse across cell membranes and they can enter cells. Many organisms produce such small molecules naturally as metabolites or signaling molecules like this cortisol that you can see here, that is a stress hormone. And these molecules often interact with proteins and influence their function. And that's why most drugs are small molecules, also because they can enter the cells through the cell membranes usually. For example, the aspirin molecule is also of this kind. And the structure of these small molecules usually allows them, because they are relatively small, allows to synthesize them in the chemical lab quite easily. Then proteins are these chain-like organic molecules that are much larger, and they are built of a fixed set of building blocks. So they are chains that fold into a defined 3D structure known as this native conformation. So it might be a bit hard to see here in this figure, but this is a chain that takes up this three-dimensional structure and then forms this globular form that you can see here by this surface approximation. And this 3D structure of the protein then also defines its function. So these proteins can be enzymes, for example, that catalyze some chemical reactions. They can be structural components, signaling molecules. And generally, they are just extremely important and basically necessary for every process involved in life, basically. And as I said, all proteins are made of this fixed set of building blocks that we call amino acids. And there are 20 standard amino acids, as you can see here on the right side. And they are the fundamental building blocks of all proteins. And they have a backbone in common. So what you can see in each amino acid is this triangle of this N, this C atom, and this C atom here. So these are basically this amino group and this carboxyl group, and they are the links of the chain. So if we connect these building blocks, these amino acids into a protein, then it's these triangles that connect and form this linear chain. And what differentiates the amino acids is just what is attached to this atom here, the C alpha atom, and that's also different for each amino acid, and it's what defines the chemical properties of the amino acids. And another fact that shows how important proteins are is that our DNA encodes for the precise sequence of proteins, of the amino acids in proteins. So each triplet of DNA nucleotides encodes for a specific amino acids. So when this DNA is transcribed to RNA and then it's translated to a chain of amino acids. And once this chain here is synthesized, it starts folding into this more ordered three-dimensional structure, which defines the protein's biological function afterwards. And this folded structure is defined by the sequence of the amino acids alone. So the folding is kind of a spontaneous process of energy minimization that is guided by these interactions between the amino acids and their side chains, such as hydrophobic interactions or hydrogen bonds. And then in a protein structure, we have these three structural levels. The first is just the primary structure that refers to the sequence of the amino acids in this chain. And then the secondary structure describes these regularly repeating local structures that are stabilized by hydrogen bonds. The most common examples are these alpha helices and these beta sheets that appear really frequently. And then the tertiary structure refers to the spatial relationship of these secondary structure elements to each other. Then you will really often see, yes? In the same sequence, you have different structures? No. So the same sequence cannot end up in different tertiary structures. So it will always have generally the same fold, but of course proteins are dynamic, so they don't always have exactly the same structure. So the fold is the same, but they are constantly moving, and there are some proteins that are more dynamic. They have some loops that can like change their position. So that's why there are also MD simulations to simulate the dynamics of a protein. So you might see different configurations of the same protein, but generally same sequence always results into the same fold.
The primary structures determine secondary, tertiary.
Sorry, again?
So the primary structure determines both the secondary and the tertiary.
Yes, exactly. And you will really often see these proteins in this cartoon depiction. And this is just to show the location of the backbone and these characteristic secondary structure elements. But as I said, it only shows where the backbone is of the protein. So if you would show the side chains of the amino acids, so the extensions attached to the tip of all these triangles that form the chain, then it would look a bit messier. So now let's turn our attention to the engineering of proteins and small molecules. So if we talk about engineering of small molecules, we have to know that they usually bind to proteins and affect their some biological process. And the chemical structure and the composition of the small molecules defines its function and also its binding preferences. So small molecules are usually engineered to interact with a specific target and then to modulate the associated disease pathway. So this can be to inhibit an enzyme or to block a receptor. An example would be this aspirin molecule that binds to the cyclooxygenase enzyme, which would otherwise form prostaglandins and cause fever and inflammation. So engineering of small molecules is kind of altering the size and the composition of the organic compound by like adding a different carbon backbone or change some functional group to get desired properties and binding preferences. And the engineering of proteins is all about changing the amino acid sequence to achieve a specific function or property. So in the field of biocatalysis, you could, for example, say that you want to change the amino acid sequence of the protein so that it can accept the new substrate. So for example, here, if you have this protein in cartoon depiction here, which is an enzyme, and you want it to accommodate this substrate, you might have to change these locations in the sequence so that this process can happen and this small molecule of this substrate can be catalyzed. And then in the context of therapeutics, you could think of an amino acid sequence of an antibody that you change in some way that it will bind to SARS-CoV-2 spike protein, for example. There's a couple of strategies for protein engineering. So there's rational design and semi-rational design. And rational design is just that you use very detailed knowledge of your protein, of your system, to make desired changes. Very simple. So you might know that for this enzyme here to bind the substrate, these positions in the chain are probably important. So you might think that this one here needs some charged amino acid so that this substrate can bind more efficiently. And that would be rational design. And semi-rational design would be, for example, that you just introduce sites that are probably important for a specific process, and then you just exchange the amino acids at all these sites to all possible amino acids, creating a combinatorial library. Then you try out all the variants, basically. And then there's the strategy of directed evolution, which is quite successful, in my opinion. It also got a Nobel Prize a couple of years ago. And it works by introducing random mutations. Then you get a pool of mutants. And then you test all these mutants. You pick out those that are most performant, and you restart the whole process. So it's kind of accelerated evolution. And it's quite successful in making a protein better at the function that it already has. So it usually requires a protein that is already working in some way, but you would like to improve it. So you can do directed evolution, and this is quite successful usually. But all these strategies are quite time-consuming and expensive, and are generally only suited for optimizing existing proteins. And the main difficulty of protein engineering is this vast size of the search space. So if you consider that there are 20 naturally occurring amino acids, each of which can occupy any position in a protein chain, then a protein of just 100 amino acids has 20 to the power of 100 possible sequences, right? And natural proteins are usually a bit longer than 100 amino acids, like a couple of hundred or up thousands. So that's where like AI and machine learning models come in. They are supposed to help us to navigate this search space a bit better and to narrow down the space of possible solutions. And that brings us to the first type of model, which is models that operate on sequences. So I'll just introduce the sequence representations that we use for proteins and small molecules, and then I will give you some examples of machine learning models. Okay, so protein in sequence representation is quite straightforward. It's just letters representing the amino acids in the chain. So, and then as you can see it here, and then for machine learning, these amino acid sequences are just encoded into some numerical representations with maybe one-hot encoding or some tokenization where each amino acid is treated as a separate token, like in text processing. And then in small molecules, this is a bit more complicated. What people often use are these SMILE strings. So it's a linear notation and that represents the molecular structure of a small molecular compound using characters. So as you can see here, atoms are usually represented by their chemical symbol. Single bonds are omitted, but the double bonds are depicted by these equal signs. And branches are described using these parentheses, and rings are noted using the numbers indicating their connection points. And then for machine learning, these SMILES codes are also tokenized and fixed to a fixed vocabulary. You've probably noted already, this sounds like natural language processing. And these methods indeed had like a big impact on the field because there's this analogy between the building blocks of language and those of proteins and small molecules. So like in natural language, you could think of protein sequence having their own kind of grammar, and they also contain long-range dependencies. So for example, amino acids would correspond to words, sequences of amino acids to sentences, and then protein would be an entire text paragraph, for example. And also in SMILES codes for small molecules, it's the same. It's also linear sequences that consist of a fixed vocabulary of symbols, and there are patterns and rules to it. So both these representations are compatible with NLP methods and that are designed to handle such sequences. So, and the first application of NLP to proteins was this evolutionary scale modeling, the ESM model trained by Meta. And it's a self-supervised language model trained with this masked language modeling objective and trained on 250 million protein sequences. So it works in this way that you have this native sequence, and then you change it by corrupting a small fraction of the amino acids with a mask. And then using this corrupted sequence, you encode it into numerical representation. And then this ESM model, very simplified here, translates this numerical representation into some latent space representation, some hidden state representation that allows it to make a prediction for which amino acids have been masked in the original sequence. And if this prediction is wrong, then the model is improved, and that's how it is trained. And it turns out that filling gaps in protein sequences is a very simple task, but it's not easy at all. So you can imagine that you need a lot of knowledge about proteins to be able to do that, and also knowledge about how proteins like form a 3D shape, for example, because at some locations in the 3D structure of the protein, some amino acids might have space to fit there and some might not. So it turned out that knowledge of intrinsic properties of the proteins emerges in these latent space representations without any supervision. And this knowledge is, as I said, encoded in these hidden state representations. And the authors, they showed this using orthologous groups of genes. So genes that come from the same ancestral protein, but maybe in different species. So these orthologous groups, they usually have a similar function, but not necessarily the same sequence. So what the authors showed is that the untrained transformer, if you pass these sequence where all an orthologous group is depicted by some color, and the untrained transformer just doesn't really add a structure to these sequences, right? But then the trained transformer is able to group these orthologous groups into clusters in this t-SNE two-dimensional representation here. So this shows us that the model kind of learned to group different sequences together based on their shared common function, basically. And these hidden state representations of this such a transformer model like ESM, they are used frequently for transfer learning. So for example, people fine-tune these protein language models on specific downstream tasks, even with a potentially quite small labeled data set. For example, for the applications like protein function prediction or prediction of mutational effects, and also for the prediction of the protein 3D structure, as we will see later. Yeah, and these pre-trained transformer embeddings, they are now used in many downstream applications. And first slide, for those of you who are interested, what I haven't been speaking about now is that there are a lot of chemistry language models that work the same way with this masked language model objective, but they are trained on these SMILE strings representing small molecules. And they all in some way demonstrate chemical learning, either by just being able to recover the masked SMILES tokens, or by clustering molecules by chemical features in the latent space representations, or just by improving downstream molecular property prediction performance, for example. And examples for this would be this mole former model from IBM research, or the Camberta model, but there are many, many more. So the next section of the talk will focus on a special kind of sequence-based models, namely the protein structure prediction models, like this includes AlphaFold 2, and also second version of this ESM model. And then in the end, I will also briefly touch upon these new folding models that don't just predict the structure of a single protein, but of entire molecular assemblies. But first, about protein structure. Very simply put, it just refers to the 3D coordinates of all atoms of the protein in its folded state. And knowing the protein structure is very important because it gives us some fundamental understanding of a protein. And it gives us also an idea what could be its function, because structure of a protein defines its function, right? And then for drug discovery, knowing the protein structure might, for example, help us to identify some active sites or interaction points for potential drug molecules. So you might maybe see this protein here and then detect that there is this groove with some interactive amino acid side chains that you might use to attack this protein using a small molecule drug. And to get protein structures, for many years, researchers relied on these very time-consuming methods like X-ray crystallography or NMR to get the structure of a single protein. And recently, deep learning has kind of taken over structure prediction. And this is quite remarkable because predicting the 3D fold of a protein is an extremely difficult and challenging task. And this is because these amino acid residues, they can adopt so many configurations due to the rotations around their backbone bonds and side chains that even a tiny protein has like an astronomical number of potential configurations. So the folding is also driven by these various interactions that are quite difficult to model, such as hydrogen bonds and hydrophobic interactions. And there are also these long-range dependencies. So the folding is influenced by interactions between amino acids that are far apart in the sequence of the protein, but then come into close vicinity in the 3D structure. And then last but not least, the training data is relatively scarce, although thousands of researchers have crystallized proteins for decades. The total number of crystallized protein structures is still relatively small from a deep learning perspective. And because this task is so difficult, but also so important, there was this critical assessment of protein structure prediction challenge. So it's an event that took place every second year, where research groups were invited to submit their models. And then these structure prediction models would be tested on a set of experimentally solved structures that haven't been released or published yet. And AlphaFold 2, developed by DeepMind, it won the 14th holding of this challenge. And as you can see here, it won with quite a big margin compared to its competitor models that also took part in the challenge. And the predictions on this unpublished set of protein structures that were made by AlphaFold, they were really precise. The deviation of the C alpha atoms, so the tips of these backbone triangles that every amino acid has, the deviation was only about 0.96 angstroms. So that is really precise. As a comparison, the size of a carbon atom is about 1.4 angstroms. This was really a breakthrough in the field of structural bioinformatics. And that's why I will now dive a little bit deeper into the architecture of AlphaFold 2. But it will still remain quite high level because it's a rather complicated model. But I will give my best. So the input to the model is the sequence of the protein, obviously. And then what the model does is it accesses a genetic database looking for similar sequences in other organisms. And then it forms this multiple sequence alignment that contains a lot of information about evolutionary conservation of certain sites in the proteins. And then it also uses a structured database search to look for structures of similar proteins that might already exist. And based on this, it makes a first guess of the pairwise distances of the amino acids in this protein. And then this MSA representation based on the multiple sequence alignment and this pair representation, they are passed into this evoformer model that is the core of the architecture of AlphaFold. And this updates the two representations through some attention mechanisms and convolution-like operations. And then these updated representations, they are passed into this structure module that just converts this into spatial coordinates. So it makes a final prediction on the pairwise distances between the residue pairs and also the angles of the bonds in the protein, and generally just constructs a 3D model of the protein. And it also applies this interesting concept of recycling. So that's this iterative refinement process where the output of the structure module here is passed back multiple times into this evoformer. And apparently this helps to refine the predictions by allowing the network kind of to reconsider its earlier outputs. So apparently the model benefits from this additional contextual information that it gets from subsequent layers. Training of AlphaFold works with a structure objective, obviously. So just the deviation from the true structure is penalized. And then there's a secondary BERT objective that operates on the MSA on the multiple sequence alignment. And the network is required to reconstruct some masked regions of this multiple sequence alignment representation. And then training also involves this self-distillation principle that I find quite interesting. So how this works is that there's this phase of initial supervised training that you just train on the data set of known experimentally solved protein structures using conventional supervised machine learning with these objectives that I showed above. And then once the model has achieved a certain level, you use it to make predictions for the structures of 350,000 additional sequences for which no structure exists yet. And then they mixed these experimental structures and the predictions of this preliminary model and trained the model from scratch using this mix of data. And apparently that makes such effective use of unlabeled sequences and makes the training dataset so much more diverse that this overall improves the accuracy of the model, which I find quite interesting. Is it okay if we take a break now and then continue with the next model after the break? Okay. So are you ready to continue? Now I would like to remind you of this model that I showed you earlier, this ESM model, the self-supervised transformer language model trained on these 250 million protein sequences. And as we said, a lot of knowledge about protein properties is embedded in this hidden state representation. And the authors also showed that while this model cannot observe the protein structure directly, and the structure is kind of like a hidden variable that influences the pattern that the model sees in the sequences. So they showed that this unsupervised pre-training also encodes some secondary structure into the representation. And they did this very easily. They just took a data set of proteins with a known structure and added labels to each amino acid, indicating whether they are part of a helix or a strand or a coil. Here you can see this in the corresponding colors. And then they just derived this transformer latent space representation for each of these proteins or for each amino acid of these proteins. And then they fit a logistic regression classifier to predict the secondary structure membership from this hidden state representation. And they get a high accuracy from this, showing that the self-supervised training indeed generates and embeds structural knowledge in this transformer representation. And that's why in a second version of the model, the ESM model is extended with a folding head to predict atomic coordinates. And they use this pre-trained ESM model based on the masked language model objective, and just add this folding head that extracts atomic coordinates from this hidden state representations. And then they train this and they fine-tune it end to end on about the same training data that AlphaFold used. And like all the experimentally determined structures from the protein data bank. And then with this, they achieve the structure prediction accuracy quite similar to AlphaFold. And this is remarkable because this strategy kind of removes the most costly aspects of AlphaFold. So for example, this multiple sequence alignment, and that speeds it up about 60-fold, according to the authors. And it's also completely standalone. So it doesn't need to access databases of known structures or evolutionary databases with related sequences. So it just takes the protein sequences and then outputs the structure of it. And then if you wonder what kind of model that you want to use for your protein structure prediction, you can see like an example comparison here between crystal structure, an ESM prediction, and an AlphaFold prediction. And we have to split this evaluation in backbone atoms, so the triangles that are the links of the chain, and in side chain atoms, because the backbone atoms are generally easier to find. So both models perform well, but AlphaFold is often still leading a bit in direct comparisons. And then if we talk about sidechain atoms, the precision of both models drops a bit because these sidechains are very flexible and therefore harder to predict. But AlphaFold generally performs a bit better in modeling the positions of these sidechains. So if you have some application where high accuracy in backbone and sidechains is crucial, then you would probably use AlphaFold. But ESM fold is nevertheless super valuable, especially if you need quick predictions, maybe for a vast number of protein sequences. But generally, protein structures are now much easier to obtain. I would like to demonstrate that briefly. So the authors of the ESM paper, they have just decided to fold 770 million proteins, even proteins that maybe don't even exist in reality. So they made this metagenomic atlas where you have all these proteins in this two-dimensional representation. Then you can explore this here, and every point in this cluster represents a protein. And the yellow ones are proteins that were previously unknown, and the blue ones are proteins that were maybe already characterized a bit earlier. But this is very impressive that they just, the model, they just used it to predict 770 million protein structures, and you can look at each of them and download the PDB file, the structure file for it. Now that we have covered the two protein folding models, I would like to move on to some recent development of the co-folding models. So models that don't just predict the structure of a single protein, but predict the structure of an entire assembly. So this could look like this, for example. So as you know, proteins, they rarely act alone. They usually form complexes with other proteins or small molecules, DNA and RNA. And recent folding models, they also include these biomolecules, these others, and form kind of a unified structure prediction tool that predicts this entire assembly with nucleic acids or metal ions or also small molecules. So examples for this would be Rosetta fold all atom and AlphaFold 3. So this Rosetta fold all atom is this 3-track neural network developed by the Baker Lab in University of Washington that predicts these structures of biomolecular assemblies. So it's trained now on a mixed data set of biomolecules, including also protein small molecule interactions or protein metal complexes, or even protein DNA interactions, right? So the input to this model is not just the protein sequence anymore, but also a potential DNA sequence, some graph representations of small molecules, or also the chemical element type of a non-polymer atom. And then this network fuses all this information and outputs some potential structure of this interaction between the entities that you added to the input. And AlphaFold does a similar thing, and it claims to have a bit higher accuracy than Rosetta fold all atom. And the differences to the AlphaFold 2 architecture that I've showed you before is that the input, of course, now includes ligands and nucleic acids and metal ions. But this multiple sequence alignment path is quite de-emphasized. So it's a much smaller MSA module. And then importantly, this structure module of AlphaFold 2 is replaced by a diffusion model. So the diffusion module now predicts the atom coordinates from this pair, single, and this input representation here. Okay, and here as well, for those who are interested, there are a lot of these co-folding models developed at the moment. So these folds models are quite famous, developed by researchers at MIT together with recursion. They are also diffusion-based, as is this Chi-1 model, also predicts molecular complexes. And then there's also the OpenFold Consortium, which is quite interesting because it's a non-profit AI research and development consortium that aims to develop like an open ecosystem of AI tools for biology and drug discovery. They also have this OpenFold 3 co-folding model. Now that we have discussed the structure prediction tools, we can transition to models that leverage these structures as input data. And in this context, I will briefly mention 3D GNNs, 3D convolutional neural networks, and then also GNN models. But before I can talk about the GNN models, I was asked to give an introduction to graphs and graph neural networks. So that's what I will do before we touch upon the GNN models. So, but first, the importance of structure. So as you know, the structure of a protein determines its function. So the proteins, they fold into these complex shapes and interact specifically with other molecules. So a protein sequence alone doesn't provide information about the actual spatial arrangement of the atoms in 3D space. So consider you have a task that requires an understanding of the physical placement of residues. Then models that incorporate this structural information should actually be superior. So for example, in interaction prediction or enzyme activity prediction. And I guess you all agree with me that if you want to make a prediction on whether this enzyme can bind or maybe catalyze this substrate here, the 3D placement of all these protein residues in this pocket should be quite important or quite useful, at least for the prediction of this model. But there are also a lot of challenges in the use of structural data. So its availability is generally much lower, and the sequence-based methods are usually simpler and more computationally efficient, right? And as I said, I will briefly explain the 3D GNNs. So how these models usually work is that they take, for example, a protein structure like here, and they convert these atomic coordinates in a format suitable for 3D GNNs. So the space around the protein is just divided into a grid of so-called voxels, and then different atomic properties of the underlying atoms, they can be used to define multiple channels in the input data. For example, the atom types or charges. In this example here, you have a carbon channel here, and an oxygen channel, and a nitrogen channel. And then you have this convolution and pooling layers to extract the relevant features and reduce the spatial dimension of the input. And then in the end, you have a fully connected layer that derives your prediction, whatever that would be. And this 3D GNNs, they have shown to perform quite well in tasks such as protein thermostability prediction, and also in protein ligand binding affinity. So this is about when you have a complex of a protein and a small molecule, you want to predict the interaction strength, so basically, which is called the binding affinity. And the 3D GNNs, they do this by centering a grid around the geometric center of the ligand, and then they each position of the atoms within the binding pocket is kind of mapped to one of these voxels, and the chemical properties are then used to define additional channels. So this model here seems to have a channel for aromatic rings, for example, and then one for hydrogen donors and some other one. And then again, like convolutional neural network reduces this 3D grid into a single PKD value that represents a binding affinity. And in this task, the 3D GNNs were the first structure-based models that were shown to outperform sequence-based affinity models. But there are a couple of drawbacks on using these 3D GNNs. So the model size, they are usually larger than 2D GNNs because the parameters, they grow cubically with the resolution of the grid. And the sparsity is also an issue because these grids that you put around proteins, they very often contain a significant portion of voxels that contain only empty or homogeneous regions of the complex that does not contribute any meaningful information. And there, the inefficiency arises because all these meaningless grid points are still processed by the model. And then there's the problem of the rotational invariance. As in 2D GNNs, if you want to achieve invariant predictions, regardless of the orientation of the input structure in the input data, then you need to present that object in several rotations. So 3D GNNs usually require larger training data sets to work well and avoid overfitting. And training and inference usually require a lot of memory and processing power. So the application of the 3D GNNs to very large data sets is limited. For example, if you have a molecular dynamics simulation and you would like to process each frame of that simulation, then you might profit from something that is a bit more efficient. And with this in mind, I would like to transition to the introduction about graphs and graph neural networks. And because these can solve most of these drawbacks of the 3D GNNs. So I will start at the very basics. The graphs describe just a set of objects that we call nodes and the connections between them that we call edges. This would be an example here. And then you can store information in the nodes of the graph or also in the edges of the graph. And then you might also have some global information attached to this entire graph. And this is quite a powerful representation of data, especially for naturally like non-Euclidean and unordered data. So if you think of a social network with friendships or citation networks, or also like atoms in a molecule. And graph neural networks, they can process such unordered graph structures. So as I said, we have these 4 types of information that we want to use to make predictions on this graph. So we have the connectivity that contains a lot of information by itself, and then the features in nodes, edges, and also some graph level features. And molecules are quite convenient to model as graphs. It's a natural abstraction where the atoms of the molecules are translated to nodes and the bonds of the molecule are translated to edges of the graph. And then it also allows for integration of chemical properties into the node, edge, and global features. So you could take this caffeine molecule here and model it as a graph like this, and then you can, for example, add some chemical atom properties into the node features because the nodes represent the atoms of the molecule. And then you can add information about the chemical bonds to the edges. And then in the global context, you could also have whatever information you have about this molecule. Yep. And then what kind of tasks can we, or do we maybe want to solve using graphs? So there's graph level tasks, obviously, that you just predict a single property for an entire graph. So for example, you have this molecule, you model it as a graph, and you want to know, is this molecule an antibiotic? That would be a graph level task. And then there's node level tasks where you predict some property for each node in the graph. What I show here is just this famous toy data set where you have this karate club and the 2 instructors, they have an argument, and then the edges, they represent the social interactions between the members of the club, and the task is in this toy data set to predict which of the members stays loyal to which instructor. And this is a node level task because we predict one property for each node in this graph. And then there's also the edge level tasks where we predict the property or the presence of an edge in the graph. And an example for this would be the friendship suggestions on Facebook, for example. And the GNN is basically an optimizable transformation on all these attributes of the graph. So on the nodes, edges, and this global context, but it preserves the graph connectivity, right? So they usually adopt this graph in, graph out architecture. They accept the graph as input with information loaded into its nodes, edges, and maybe global context. And then they progressively transform these embeddings without changing the connectivity of this input graph. And to update these graph features in the nodes and edges, they usually apply this message passing neural network framework, where neighboring nodes and edges exchange information to influence each other's updated embeddings. So this makes the learned embeddings, kind of aware of the structure of the graph and of the neighborhood of a node, for example. And this message passing works in a way that for each node in a graph, we try to gather all the neighboring node embeddings or messages, like here, and then we aggregate all these messages using an aggregation function. In this example, it's just summing them up, but it could be other ways. And we also include the embedding of the node itself. And then we pass these pooled messages through an update function, which is usually a learned neural network, and then we obtain the updated embedding of this node. So from this perspective, from the perspective of this single node, we collect all these node embeddings of these neighbors it's connected to, we aggregate them, pass them through this learnable weight matrix, and then we have this updated node embedding, and we do this simultaneously for all nodes in the graph. So, and then if we want to take predictions, then for example, for a node level, then we just take all the node embeddings and pass them through a fully connected neural network to get some predictions for each node, and same for edges. And if we want to do a graph level prediction, so for example, is the molecule an antibiotic or not, then we can use global pooling, for example, to pool all node embeddings into one single representation and then pass them through a neural network, and that makes a prediction. Or we can just process the transformed global features of the graph. So this has a lot in common with the image convolution. Both are operations that aggregate and process the information of an element's neighbors to update the element's own values. So in graphs, these elements are just nodes, and in images, they are pixels. And if you consider a graph where each node is connected to its neighboring nodes and it's arranged in this 2D array, then it would be quite similar to image convolution. But in graphs, generally, the number of nodes in the graph is the number of neighboring nodes is quite variable, and also the structure is of course not adhered to, so there's they can be very unordered, right? And that's why, because of this analogy, the message passing process in these GNNs is also called graph convolution. And then, as in 2D CNNs with images, for example, we can also stack graph convolutional layers. And then if we have 3 convolutional layers all doing message passing, then every node will contain information about every node that is up to 3 steps away from itself. Then there are a couple of, or quite many different convolutional operators. So one of the most common and most easiest is this GCN-conv, where you just, you want to find the updated embedding of this node I in the next layer. And for this, you go through all the neighbors of this node I. You take their node embeddings, you pass them through a learnable weight matrix, and you normalize by the degrees of the nodes. Then you sum this up to get the new embedding of this node I. And in this attention version, it works quite similarly. You also take all the neighbors of this node I, their embeddings, you multiply them with this weight matrix, and then also an attention score. And this is computed based on the embeddings of the 2 nodes and the edge between them. So from the perspective of a single node, you would kind of assign importance values or weights to all this node's neighbors. And this is then taken into account in this aggregation function. So now that we have established these GNN basics, we can proceed to these examples of GNN models. They were mainly for drug discovery. And The first very cool model is this GNN that is capable of predicting molecules with antibactericidal antibacterial activity. And this is a graph binary classification problem. So you have a molecule, you model it as a graph, and you want to get one prediction for the entire graph. The training set for this model was only about 2,500 drug molecules, and among them, 120 of them are known antibiotics. And then they generate these graphs of the molecules, and they include a lot of chemical properties into the nodes and also into the edges of the graph. And then they do a couple of layers of this convolutional layer, so of graph convolution, followed by global ad pooling, and then they have a feed-forward neural network that just outputs a probability of growth inhibition of E. coli, so of antibacterial activity. And this model is quite simple, but they then used it to make predictions, so inference on a large collection on the drug repurposing hub, a large collection of small molecules. And then they saw this really nice enrichment that among the 99 molecules with the highest model prediction, 51 of them actually displayed growth inhibition. And among the bottom 63, they only had 2 molecules that displayed growth inhibition. So you can see that here, if you have a certain concentration of bacteria, and then you add this halicin, the new antibiotic that they discovered, you add this to the mix, and then these bacteria start to die. So they also showed that if they pre-treat the bacteria with, what was it? Ampicillin, quite an old antibiotic that doesn't work that well anymore. They can even kill the persisters, so the bacteria that resisted to the ampicillin. And then they also saw that this new antibiotic had quite a low structural similarity to its nearest neighbor antibiotic. So they concluded that the model was kind of able to generalize and access some new antibiotic chemistry, which I think is quite remarkable for this model. And then another example of graph neural networks, they are really often used for this prediction of binding affinity, so the interaction strength between a small molecule and the protein, using the 3D structure of this interaction as an input. So this is really important for drug design, because you want this small molecule to bind with a really high affinity to very specific proteins so that it can be a useful drug. And the models that take such a structure and assign a binding affinity, they are referred to as scoring functions. And classically, these were force field-based or empirical or knowledge-based models, but unfortunately, they show quite limited accuracy in binding affinity prediction. And AI models are now supposed to improve that. And an example for a model is this GEMS model. It's quite simple and easy to understand. It's also a graph convolutional neural network for predicting these binding affinities, and it's trained on PDB-Bind, which is a collection of experimentally solved protein ligand complexes that have binding affinity labels. So if you have this interaction here of this small molecule with this protein, then the small molecule is included as an atom-level graph of this ligand, as we've seen before. And then the surrounding amino acids of this protein are included as additional nodes. And then in this model specifically, the node features of the graph were initialized using protein language model embeddings. And the architecture is quite simple. As an input, you have these node features, the edge features, and some global features, and of course the connectivity. And then there's after an initial feature transformation, you have this alternating sequence of node feature updates and edge feature updates. So the nodes are updated based on their neighbors, including the embeddings of the edges that connect them to these neighbors. And then the edges are updated based on the nodes that they connect. And this happens like twice. And after every node update, we have this global pooling that updates the global features, like the global context matrix. And then in the end, we have this simple feed-forward neural network that reduces this to a final prediction, to a scalar value that represents the binding affinity prediction. And the prediction of this model was quite more accurate than the classical scoring function I mentioned. And it also showed that if you add these embeddings of the protein language models to the node features, this boosts performance. And also if you add embeddings of several protein language models, this kind of has a synergistic performance improvement. Yeah, which is quite interesting and also shows how these protein language model latent space representations can be useful for downstream tasks, right? So we are at the conclusion for graph neural network section. So they are the GNNs, they offer a lot of advantages for modeling and processing these 3D objects. They can handle the data that is defined in these irregular domains without mapping them to a grid. And they are naturally invariant to translation and rotation. And they are also much sparser representation of a protein or a molecule than these 3D representations in the GNNs. There's also many possibilities for data integration and this efficient update mechanism with graph convolution that also uses weight sharing. And so GCNs, they usually perform quite well in tasks where an understanding of the structure and the interconnectivity of the data is important. And they also usually perform quite well with a relatively low number of parameters. Yes, do you have any questions? Next week, we will first talk shortly about some challenges in training models on biological data, and then dive into the topic of generative AI for the design of such small molecules and proteins. Thank you for your attention.